Es para mí un honor presentar al profesor Sir Mark Caulfield. Haré una breve reseña de su biografía. El profesor Caulfield se graduó en medicina en 1984 en el London Hospital Medical College. Formó parte del NICE Guideline Group para la hipertensión y fue presidente de la British Hypertension Society entre 2009 y 2011. Fue director del Instituto de Investigación William Harvey entre 2002 y 2020 y fue elegido miembro de la Academia de Ciencias Médicas en 2008. Desde 2008 dirige la Unidad Cardiovascular y de Investigación Biomédica del National Institute for Health Research, BARTS. Entre 2010 y 2015 codirigió la fusión de 3 hospitales en el norte de Londres para crear el nuevo BARTS Heart Center. Ha ganado el premio Lilly de la Sociedad Británica de Farmacología, el premio Björn Folkow de la Sociedad Europea de Hipertensión en 2016 y el premio Frank Borchardt de la Sociedad Internacional de Hipertensión en 2018. En 2013 se incorporó como investigador senior en el NIHR y fue nombrado director científico de Genomics England, del proyecto de los 100.000 genomas. Sir Mark recibió el título de caballero en la lista de honores del cumpleaños de la Reina de Inglaterra en junio de 2019 por sus servicios al proyecto 100.000 genomas. Hoy nos va a presentar la conferencia titulada 100,000 Genomes Project, Transforming Healthcare. Sir Mark, thank you very much for having accepted to give us this conference. It is a pleasure for all of us to have you at this translational research meeting today. Please go ahead with your presentation.
Carmen, thank you so much for having me here. I'm deeply honored. I'm sorry we can't be together in person, but one day we'll be able to do those things again. So, ladies and gentlemen, it's a privilege to be with you, even if it's over the internet. I'm going to share with you a little bit about the 100,000 Genomes Project focused on commoner disease, and also some of my own work in blood pressure genetics of cardiovascular disease, and how I think that can be an example of how we move forward with common complex disorders. I'm also going to touch on our latest findings in pharmacogenomics and share with you the work that we've been doing to understand the susceptibility to severe COVID during the pandemic. So these are my disclosures. Currently I'm seconded to a government company called Genomics England from my university, the William Harvey Research Institute at Queen Mary University of London. So I thought I'd start with some ancient history. In 2023, my hospital, St. Bartholomew's Hospital, where I work, will be 900 years old. So it's one of the oldest hospitals in Europe. And during the 1600s, from 1609 to 1640, a famous physician who trained at Cambridge and in Padua, William Harvey, made his observations and wrote his book on the discovery of the modern view of the circulation, which we still use today. What you can see on this slide on the left-hand side is the record of a meeting of the governors of St. Bartholomew's Hospital from March 1639. And you won't be able to read any of this. And even if you could, it's written in Old English. So I'll translate some of it for you. It is hard to read, but I will explain some important things. So this was the 23rd of March, 1639. And this is a proposition to the governors for a scurvy grass drink by Dr. William Harvey and others. There are some slightly worrying ingredients here, such as 8 gallons of beer, but you'll notice in blue that there is scurvy grass, there's watercress, there's horseradish root. So some 60 years before James Lind did the randomized trial showing that fruit, which we now know contains vitamin C, William Harvey had proposed a drink containing the key elements that could reduce scurvy. So I just thought I'd bring you that little bit of history before we start. Now, meanwhile, back at the genome, you and I have 3.3 billion letters in our genetic code, and there's approximately a variation about every 300 letters or so If you add up rare and common variants, we mostly have between 5 and 6 million variants genome-wide. And they can be single letter chains, single nucleotide variants, extra bits and then missing bits, insertions and deletions, copy numbers, structural variants. And we've been able to begin to access these with large-scale genotyping and more recently, increasingly with next-generation sequencing. And this is paving the way for major discoveries in rare and complex disease. So I was originally involved in the Wellcome Trust Case Control Consortium in charge of the blood pressure element of that consortium, and comparing 2,000 controls with 3,000 cases, we didn't find a single locus for blood pressure. And many people at that time wondered whether we should continue to look for the genes for blood pressure. I hope I'll be able to convince you it was worthwhile when you see the results in a moment. So the UK has a government strategy called Genome UK for genomics in healthcare. And it categorizes the assets that we have in the UK into diagnosis and personalized medicine within the healthcare system. So that's Genomics England and the NHS, where we're predominantly working on a rare disease, cancer, and infection. And we've sequenced over 111,000 whole genomes from 90,000 people. And over the next 5 years, we will sequence 500,000 whole genomes from people with rare disease. And that will become part of a national genomic library. That's whole genome sequencing. There is a new cohort of 5 million people proposed, which has not started yet. This has been called Accelerating Detection of Disease, but is about to change its name to Our Future Health. And that will be 5 million representative people across the population. And this will look at the role of genotyping and polygenic risk scores. And of course, we have the asset which many of you work on, which is UK Biobank, which is 500,000 whole genomes and now has several 100,000 exomes. And from the end of this year, there will be 250,000 whole genomes. The final study is the National Institute for Health Research Bioresource, which is aiming for 200 to 400,000 participants that are disease-focused, where there will be whole genome sequencing or genotyping. And so we're looking to join up these national assets so we can have both the scale and the content alongside rich clinical data assets to be able to make real advances in genomics and mobilize that into healthcare. So I'm going to focus a little bit on blood pressure genomics. This has been an area of my interest now for over 30 years. And so just to give you a little bit of background on that, and I'm using this really as a model for common complex disorders. So blood pressure is the most important risk factor worldwide. It's important in 77% of stroke, half of coronary disease, and it's estimated to have caused 10 million deaths in 2017. By 2025, there'll be 1.5 billion people worldwide who have high blood pressure. That's an enormous proportion of the population. And control of blood pressure is still a challenge. So we know a little bit about the genomic architecture of blood pressure from familial studies. And we think that about 30% of blood pressure variation is due to genes and their interactions with lifestyle. Things like being overweight, consuming excess alcohol, adding excess salt to your diet, lack of exercise, and failure to eat a green leafy diet. So these are the lifestyle factors. We know from work, very elegant work done by Rick Lifton, Xavier Jean-Maître, and others around the world, Frederick Luft, that there are rare syndromic forms of blood pressure and that these can tell us about therapy. So what I'm going to share with you now is some of the progress we've made with blood pressure as a complex trait. And this is using genotyping, where we might genotype a chip of 1 million variants and then use it to impute between 7 and 9 million variants based on our knowledge from the Human Genome Project of the architecture of our genome. So I'm just going to share with you a project that I led and published in Nature Genetics in 2018. This is a genetic analysis of over 1 million people, which identified 535 new loci that were associated with blood pressure traits. So what we did here was to use the population of UK Biobank, combine that with studies of an international consortium of multiple different studies called the International Consortium of Blood Pressure Genome-Wide Studies. to yield a discovery population of 750,000. And then we had both a two-stage and single-stage analysis where we worked with the Million Veterans Program, 220,000 people, and the Estonian Genome Project with 28,000 people. In total, this population added together yielded a group of over 1 million people. So where we did discovery in 750,000 and validated it in the Million Veterans and Estonians, we discovered 325 novel loci. But when we looked at the Manhattan plot, we could also see many loci that were above the threshold for genome-wide significance. And there's been an increasing trend with as populations have enlarged and a validation population isn't so easy to find to use a single stage analysis. So what we did was we set a really stringent p-value of 5 times 10 to the minus 9 for accepting a discovery and evidence in favor of replication in other resources, in each of the resources. So we found another 210 novel loci from this approach. So this is the largest number of loci discovered for blood pressure in a single study. This is the Manhattan plot. Now, if I share with you that when I looked at my Manhattan plot from Nature in 2007 for hypertension, there was nothing even approaching genome-wide significance. And now here you see for systolic blood pressure and diastolic blood pressure that there are multiple signals passing not just 5 times 10 to the minus 8, but also 5 times 10 to the minus 9 thresholds for genome-wide significance. So this provided an extensive new vista on the biology, and we combined all the known and new variants at different loci to yield a polygenic risk score that predicts an odds ratio of up to 3.5 for hypertension risk and a potential 13 millimeter change in blood pressure across all the deciles in the over 50s. So imagine that this could be, with further research, an opportunity to undertake a genotyping chip in early life, work out your polygenic risk score for blood pressure along with other traits, and perhaps intervene with lifestyle to try and avoid that. I don't know yet whether that will work, but with scale studies like Our Future Health at 5 million and UK Biobank, we can begin to tackle these challenges. And this is what, of course, ladies and gentlemen, makes this relevant for healthcare. Importantly, we discovered some pathways that are worthy of further study because they have clear biological links to blood pressure. So urotensin, which causes profound vasoconstriction through the urotensin 2 receptor, relaxin, which causes vasorelaxin, and RAMP2, which is a locus for the adrenomedullin receptor. And then drugability analysis showed that at the SLC5A1, the canagliflozin molecule and other gliflozins might be targeting a genetic mechanism for blood pressure, presenting a potential repurposing opportunity. Now, of course, many of you here who know of these drugs used in type 2 diabetes, where they encourage you to secrete salt and sodium salt and also sugar, are already known to cause a lowering effect of blood pressure. But here you have a potentially causal association. It's not proven, but a potential mechanism that you can target that may underpin the disease. We explained with these 1,284 known and new loci, 7% of blood pressure variance. So in a second paper that was published just at the end of 2020, there was a new focus in 1.3 million individuals on rare and common variants. So the focus to date have been on variants above 5% frequency in the population. This study looked at rare variants and found 106 new blood pressure associated genome regions and 87 rare variants that affect blood pressure. 32 of these were entirely new blood pressure loci. In other cases, they were new variants in known loci, and 55 are independent of blood pressure association variants within the known regions. The average effect size for rare variants, these are ones less than 1% frequency, was 8 times larger, and I'll show you the impact of that in a moment, than common variants, indicating potential candidate causal genes. And you can see here on this slide that there were several protein coding region gene chain variants, missense variants, changing proline to serine in this gene ARH-GAP31 that causes Adams-Oliver syndrome. And other Adams-Oliver genes also are associated with blood pressure, and a rare variant in the GATA5 gene, which is known to be associated with congenital heart defects. So this, for the first time really, ladies and gentlemen, shows us strong evidence that there will be rare variants, and this may explain some of the missing heritability for blood pressure. So recently we published just in the last month in Hypertension this review, which you might like to look at, by McGovern et al. And what this review shows is the different studies building up from left to right. And what you can see is the increasing shift from merely a focus on common loci and the inception of a single stage discovery in the dark blue to red on the right-hand side, which is where rare variants are being studied. Let's take a look at what the difference of the effect size between rare variants and common variants. So on the right-hand side of this plot, you're seeing in black, the systolic blood pressure, in a red diamond, the diastolic blood pressure, and in a triangle, pulse pressure. And so what we've done is arranged from left to right, the variants and their effect size. So as you come over towards 0 on the bottom line, you are in the rare variant space, and you can see that there are effect sizes above 16 millimeters of mercury in some cases, and that explains the 8-fold increase in variant effect size for rare variants. And this is what we would expect, because these may be behaving a bit more like a simple Mendelian trait and a monogenic disorder. I'm going to move now to whole genome sequencing in cancer and share with you an update from when I spoke to you before about this. So we've worked on 17,488 patients with cancer using fresh tissue genomes because we found we couldn't make formalin work. And here we're comparing the whole genome sequencing of your germline genome, the genome you inherit from your mother and father, and the somatic genome of the tumor in fresh tissue samples. And you can see here the different cancers that we've worked on. So in this slide, I'm showing you data from 5,700 people across the common cancers that you see at the bottom. And this is really to say up the side here, you're seeing tumor mutational burden. And you can see for example, for endometrial cancer, colorectal cancer, lung cancer, and melanoma, an increased tumor mutational burden. And people with the increased tumor mutational burden may preferentially need to go down an immunotherapy route at an earlier stage because they may be declaring a risk of having an adverse outcome. So here's a patient that we've recently worked up in the last 2 years. This is a gentleman aged 42 who presented with a colorectal cancer deposit. And on the right, you're seeing a CT scan, a section through the liver. And what this is showing you in dark gray is a substantial tumor deposit in the right lobe of the liver. Imagine that you're looking at this person's liver from their feet. And so usual care testing revealed no genomic markers that could stratify healthcare. So he received usual care. The lesion enlarged, his liver was getting larger, he was in pain. We did a rebiopsy, did a whole genome sequence, and it turns out that he has mismatch repair and Lynch syndrome. He's a late presenter with Lynch syndrome, and he has gone down an immunotherapy route to illustrate that. So this is an example of where the whole genome sequence in a common disease is providing access to a stratified healthcare opportunity for patients. This won't be curative, and all of you know that because of the extent of disease, but what if it could buy some good quality life for a period of time? I wanted to just share with you what the health system did with what our returned results. We returned results. This is one centre in the middle of England, West Midlands, and they had 3,244 patients on the left-hand side. If you move to the central column, you can see that in orange listed for GTAB, this is the genomic tumor board, and 781 of the patients went back to the tumor board after their genome was sequenced. And of those, 477 had a clear action, that's 61%. And you can see here a range of pharmacogenetics with DPYD, a change in the clinical use of the medicine for licensed action, a change for unlicensed action. In other words, they got a medicine that wasn't licensed for their tumor. 204 got into a trial, 43 of high tumor mutational burden and may get an immunotherapy, and 69 were referred to clinical genetics. It's important also to point out that there were 1,416 patients for which no clinical further action was considered at that time. But ladies and gentlemen, their genome is banked and ready and there if they do recur in the future, so that we can use genomics potentially to profile the best therapy for them. I'm going to dwell a little on pharmacogenomics. So we've looked in Genomics England at the Clinical Pharmacogenetics International Consortium Consensus Guidelines, and we've looked across now 60,000 whole genomes. And what we find is that 100% of people that you look at possess a CPIC actionable gene-drug pair. By that I mean they have at least one gene variant that predisposes them to either ineffective therapy or harm. So we've enlarged that to 80,000 now, and we see the same picture. So it looks like almost all of us have a potential pharmacogenomic interaction. And I can tell you that across 60,000 genomes, there were a median of 4 gene-drug pair interactions. So we're preparing now in the NHS to bring live panels to test for these drug interactions, where potentially response mode pharmacogenomics may explain an adverse response, or indeed preemptive pharmacogenomics, where we test people before we give them the therapy because the potential harm is such a risk. So to make the data that we've got in the 100,000 genomes come to life, we've invited all of you to join us as researchers, and over 3,000 from 33 countries have today, and they've won 50 million in grants, and we also work with companies. I'm going to just speak finally about our human COVID program. So in the UK, we have a human COVID program comparing up to 20,000 patients that are severely affected with 15,000 controls. And we're sequencing them with whole genomes. We've published our first paper in Nature in December. I'll show you the data. And we also have other data that we can bring to bear as controls from the whole 100,000 Genomes Project. And the outputs could be, we are mapping every patient to our viral sequencing program. So we'll be able to look at the viral genetic makeup, the human genetic makeup. Is there something that's human? Is there something that's viral? Is there something between the two? And that's hardly been done before. And the outputs could be understanding better the architecture of the pandemic, because this looks like it'll be with us for a while. We've also shown that you can identify potential therapies for severely ill people, and I'll show you those in a moment. And possibly there may be the ability to introduce risk scores for future pandemics. So I'll just move to the COVID-19 data. And this is genotyping chip data, because we could do that most quickly, on 2,224 patients published in Nature in December. And you'll see that 30% are women, the average age is 57, 25% are from minority communities, and 22% have sadly died. Here is the Manhattan plot that we found. Now, this is quite unusual, arranged chromosome 1 through to X on the right-hand side, and the p-value up the side, with the dotted red line being genome-wide significance. So we replicate the locus found in Spain and Italy on chromosome 3. We didn't replicate the ABO blood group locus, and we think that that locus contains a chemokine receptor 2, which is responsible for migration of white cells into inflammatory regions, and that might explain the white cell infiltrates in the lung. We also find tyrosine kinase over here, interferon, and DPP9 is the locus for idiopathic pulmonary fibrosis. So there are several loci here which may produce new vistas on the biology of severe COVID. So how have we used these? So we did a druggability assessment, and we found that there were 3 potential therapies. Interferon, of course, is trialed in severe infection, has not succeeded so far, but we're bringing live, and there has been a publication in New England Journal on baricitinib suggesting improved lessening ITU stay, and baricitinib is coming to a limb of the recovery trial. And we're also looking at chemokine receptors, which are biologics, which are used in rheumatoid arthritis and psoriasis. So I thought I'd finish with, we really have strong public and patient engagement, because the success of genomics is about whether we can bring our patients with us and they trust us and believe we're doing the best thing. So I wanted to show you a survey from 2019, which we did in 1,800 adults. And this is about asking the question of people who've not had a genetic test for any reason. What do you think about genomics, and what would you like from genomics were you to be able to have access to genomic testing? And 72% said they'd be interested in pharmacogenomics, 71% said they'd be interested in current medical conditions, healthier life is 68%, and inevitably some people are interested in their ancestry. So it's quite interesting to see this because it affirms a growing interest and also a growing knowledge base that we see in the UK amongst the public, and an appetite to see this benefit them across their lifetime. So what is Genomics England doing over the next 5 years? Well, we're just about to be announced whether we've been refunded by the government. We have a program for diversity in pharmacogenomics because people from minority communities are underrepresented in genomics research. We're continuing our work on cancer because the job is not done. We will work with others on polygenic risk scores, but our most innovative program is this one, which I'm really pleased the government have adopted in their 10-year strategy, which is to look at newborns, and it's called Generation Genome. And the idea behind this is to look for approximately 600 conditions that in the first 5 years of life cause severe harm, but there is an intervention that might delay, avoid, or prevent that harm. And that affects 1 in 190 births, and there are 9 children born every day in England who have these disorders. So these are known fully penetrant rare diseases where we could intervene. The interventions are mostly simple. They're diet, vitamins, minerals. 8% are expensive medicines, and some are organ transplants, which are more expensive. But if you look at the economics of these disorders, they're hugely costly over a life course. And so anything we can do to improve the life of these people has got to be something which we will test with research, with ethical and societal debate throughout. So ladies and gentlemen, in conclusion, I've shown you that in common complex traits, we've made real traction in several of them. I'm a cardiovascular researcher, so inevitably I've shown you exemplars from blood pressure, but I could have shown you other traits. And it's amazing to see how that's changed. It's also bound the worldwide global genomics community together. And that's been a great force for good. I can now say for sure that rare variants will also be important, and it could be that this is part of missing heritability. I think other omics, for example, epigenetics, post-translational modification, etc., these may all explain some of the missing heritability. In our recent paper in December, we showed Mendelian randomization, causal relations of the variants that we'd found to stroke and coronary disease, and identified potential targets. In cancer whole genomes, I've shown you that we can reveal precision medicine opportunities to tailor care for cancer, and that pharmacogenetics is probably going to be relevant for all of us. And I've shown you some of our UK plans. But I firmly believe, and that's why I'm here, Carmen, that the future is a global coalition of intellects, and it's a pleasure to be amongst people of similar interests in Spain today. I should like to thank the participants in the 100,000 Genomes Project. I should like to thank all the people who made this possible, and in particular you for having me today, and happy to take questions. Thank you so much.
Thank you so much, Sir Mark, for your extraordinary conference. It is really remarkable to learn about the achievements of 100,000 Genomes Project regarding not only rare diseases and cancer, but also common diseases like hypertension, colon cancer, and others like COVID-19, and also pharmacogenetics. And also, thank you very much for, and congratulations for the ambitious future plans related with different programs like newborn screening. We appreciate very much you have shared with us your experience. With this in mind, I start the questions. Could you tell us which were the main challenges you had to face to develop genomic medicine in UK? It was organization, technical and human resources, or perhaps ethical and legal regulation?
So when we started in 2013, which is just over 7.5 years ago, the situation in the National Health Service was that genomics, you could get access to some genomic testing. It wasn't the most advanced testing, and it was very regionally different. So some regions were really in a good place, others didn't have had very little. So this caused long diagnostic odysseys for children. So when we started, the first thing to do was to win hearts and minds with the health professionals that were needed to make this a durable legacy. And so I spent a lot of time doing that. And then we needed to overcome the technical challenges you describe. So how would we sequence 100,000 genomes? How could this be done? And I can share with you, it's not a secret, there were no shortage of people who not only would not work on the program, but were very, very doubtful this could ever be done by our system. And so what we did was we looked at all the technologies in the world, we tested all the technologies, and it turned out, it won't surprise any of you, that Illumina were in the best place to deliver the program. And I have had a fair amount of experience of genetic work in the lab over the years. And what I realized is that if you have a traditional consumer-supplier relationship, it doesn't work so well because, you know, you have a problem in the lab, you don't know whether it's Illumina's reagents or your way of working. You spend about 6 weeks in discussion with Illumina, and eventually they send you some new reagents. So we decided that we would ask Illumina to do the sequencing. So they not only own the success of the project, but they also owned with us the problems and the challenges. And we worked together, and actually it's been an extremely good partnership. And more recently, we're doing similar things with Oxford Nanopore. So we have our own Promethean, and we're doing long read whole genomes in intellectual disability with France Genomics. They're doing a similar study in France, and we're combining our data. We work quite closely with France Genomics and Australian genomes. And I firmly believe, and we'd be delighted to do this also with Spain, that this is a challenge for humanity, and we should solve this together as a coalition of global intellects, not as individual nations. So you're welcome to come and work on our data. That's all fine. And then we had to embed that in the NHS. So we created Genomic Medicine Centers of Excellence. And of course, that meant at peak, 5,000 frontline staff worked on the program, Carmen. And so that means we built out the legacy. So these people now want to do this. They've been doing it for 5 years. They don't want to stop. They don't want to go back to where they were. And so we now have a national framework of genomic lab hubs and also hospitals that are engaged in the program. And we will have regional equity of access to care across 57 million people in England, and Scotland, Northern Ireland and Wales will follow. And then the analytic challenge are not inconsiderable. So not only was there really no end-to-end pipeline that really worked end-to-end, which you could flow through things, we had to build that, but also nobody had accredited such a pipeline for return of results to the clinic. So in Europe, you and I have to use ISO accreditation, but we now have the world's first whole genome end-to-end ISO accredited pipeline. When you talk to others, they have bits of it, but they don't have it all end-to-end. And that's what you need. You need almost a production line to deliver this. I'll stop there.
Thank you so much, Mark. I have another question from the audience. It has been made by Vicente Soriano, and he congratulates you for the presentation and say that if you have any clue from your severe COVID-19 genomic characterization about the split out into two groups, one, entry susceptibility, two, innate and adaptive immune response, causing either hyperinflammation versus hypoinflammation. Thank you.
So, Carmen, the answer is yes. We think that the chromosome, I mean, we don't know yet, but the chromosome 3 locus contains chemokine receptor 2, and this leads to migration of macrophages particularly. And one of the observations in intensive care in several centers worldwide has been the amount of infiltrate of white cells into the lung tissue. And it's well established now that in children, you can get a systemic inflammatory response. So that's down the inflammatory response end. Down the response to infection, it's probably unsurprising that interferon 2 is a locus. If you look, there is an international consortium which is preparing a paper for submission for publication with many thousands of patients who've been exposed to COVID, but only 6,000 have been severely ill. What they're seeing there now is an expansion to 15 loci, some of which do mediate immune response, but that's unpublished data. So I think it'll be a bit of both. The other thing I didn't say, Carmen, is that on one of the chromosomes, there is DPP9. I mentioned it as a locus for idiopathic pulmonary fibrosis. But what if long COVID is dependent on your DPP9 genotype and whether or not you have a continuing ongoing reaction in your lung of fibrous tissue, which of course makes your lungs less elastic and less functional. So I think there's a lot yet to discover, but the question's a really good one.
Thank you, Mark, for your answer. I don't see any other questions coming from the audience. I'm just giving you the thanks for your participation and also for showing us all the achievements you have had implementing the Genomes Project in your country. I hope that it will be the same in Spain in the next future. Thank you for your explanation and also for your advice.
Thank you for having me. It's been an honor, Carmen. Thank you.